Okay, coming to metabolism, choline is the most important lipotropic factor and it prevents fatty liver disease. Next, the mechanism for thermoregulation and heat loss from the body is radiation. In children, non-shivering and heat energy production in the newborn is because of the brown adipose tissue that is found in the interscapular region. Then the next table is showing from which substrate the energy is utilized or energy is taken. Like in muscle, in well-fed state, glucose is used. In fasting, fatty acid is used. In starvation, fatty acid is used. In liver, glucose is used in well-fed state. In brain also, and in heart, fatty acid is used. In fasting condition, liver uses fatty acid, and in starvation, it uses amino acids. It never uses ketone bodies. Then brain uses glucose in fasting condition also, and in starvation it uses ketone bodies. Heart uses fatty acid in well-fed and fasting state, and it uses ketone bodies in case of starvation. It never uses glucose. Next, table is showing the amount of energy we get from the metabolism of different substances like carbohydrate, amino acid, protein, ketone bodies, alcohol, and fatty acid. Next, there are some single line high yielding points like the skeletal muscles have a large storage of glycogen which provides energy during exertion or during motion. In resting muscles, they use fatty acids. Fatty acids are major fuel for the heart, renal cortex, and adipose tissues. Retina is completely dependent upon glucose as a fuel source. After overnight fasting, the level of glucose transporters, that is GLUT, are reduced in the adipose. Next, metabolism in starvation. Alanine from the muscles and glutamine from the liver are the most important source for gluconeogenesis. Ketone bodies are formed after day 3, and ketone bodies are the main source of energy for brain in case of starvation. Then ketone bodies are found in starvation as acetoacetate and beta hydroxybutyrate. Ketoacidosis without hyperglycemia and occurs in glycosuria. It is less severe than diabetic ketoacidosis. During prolonged starvation, the rate of gluconeogenesis depends on increased alanine in the liver. In a well-fed state or in absorptive phase, metabolism is from malonyl-CoA, which is found in the fatty acid synthesis or fatty acid metabolism. Coming to ketosis, it occurs in liver and is mostly adapted in case of starvation. Then ketone bodies are derived from acetyl-CoA and they serve as a fuel for extrahepatic tissues in starvation and in uncontrolled diabetes mellitus. Ketosis of starvation differs from ketosis of diabetes mellitus as ketosis of diabetes is more severe and associated with hyperglycemia and glycosuria, while in starvation ketosis is less severe and is not associated with the two, hyperglycemia and glycosuria. Next is about acetoacetate, which is most important ketone body, and it forms acetone and beta hydroxybutyric acid both. Then acetyl-CoA enzyme is starting material and HMG-CoA synthetase is the rate limiting step in ketone body synthesis. Acute liver failure with increased ketone bodies is seen in raised. In anaerobic glycolysis, the number of ATP produced is 2. In aerobic glycolysis, the number of ATP produced is 7 from malate and 5 from glycerophosphate. Okay. Coming to Krebs cycle, citric acid cycle or tricarboxylic acid cycle. It is an amphibolic pathway. Amphibolic means both catabolic and anabolic functions are taking place, that is, energy is being produced and energy is being utilized in this cycle. It is happening in all the eukaryotic cells where mitochondria is present, but function only as a aerobic metabolism when oxygen is available. Hence, it doesn't occur in RBCs as there is no mitochondria and there is no oxygen supplied to it. So, RBCs are totally dependent upon glucose anaerobic glycolysis for their functioning, for their metabolism. Coming to acetyl-CoA, that is the common product of carbohydrate, fat and protein metabolism. It is the starting material for fatty acid synthesis and it serves as a link between glycolysis and Krebs cycle, similar to pyruvate dehydrogenase, which is also a linking point between glycolysis and TCA cycle. Okay, this is the fate of pyruvic acid. So glucose is converted from aerobic glycolysis to pyruvic acid. Pyruvic acid after oxidative decarboxylation is forming the acetyl-CoA, which is forming the TCA cycle. Then acetyl-CoA is for fatty acid synthesis. It is the rate limiting enzyme and it is forming the malonyl-CoA. Then in the process of gluconeogenesis, pyruvic acid is forming OAA. OAA is oxaloacetic acid. Pyruvic acid may also, in the process of anaerobic cycle, may form lactic acid, and by amination it may form alanine. Then this is the table showing the number of ATPs totally produced. So in the process of anaerobic glycolysis, 2 ATP per molecule of glucose are produced. In aerobic glycolysis, 5 ATP by glycerol 3-phosphate shuttle and 7 via malate pathways seen. In TCA cycle, total number of ATP produced is 10. In ketogenesis, 26 ATP. In aerobic oxidation of glucose, 30 ATP via malate shuttle and 32 by glycerol 3-phosphate shuttle. By HMP shunt, remember the answer is 0. There is no ATP or no energy produced in this shunt in this cycle. Then in beta oxidation, if palmitic acid is formed, it will lead to formation of 106 ATP, and by stearic acid 120 ATP are produced. Then this is a cycle of citric acid cycle or TCA cycle or Krebs cycle. Here you can see pyruvate, which is formed in the glycolysis from these essential amino acids, is forming acetyl-CoA. These acetyl-CoA, the linking step between the glycolysis and the fatty acid synthesis or TCA cycle, is converting the oxaloacetate OAA to the citrate in the presence of citrate synthesis. Then it is converted into oxalosuccinate, which is forming the alpha ketoglutarate, and they are producing the arginine, glutamine, glutamate, histidine, and proline. Then going to the succinyl-CoA, which is an irreversible step, the arsenase present here will be forming the alpha Kg dehydrogenase complex. Succinyl-CoA is going to act in the presence of methionine, isoleucine, threonine, and valine. Here the ATP produced in the Krebs cycle. Then succinate is leading to formation of fumarate, then malate. Then again it is going to the formation of oxaloacetate. Then this table is showing the comparison between glycolysis, citric acid cycle and HMP shunt. Remember the number of ATPs produced in the cycle total. In glycolysis, the end product is pyruvate and lactate. In citric acid cycle, carbon dioxide and water is the end product. And in HMP, glycerol 5-phosphate, 6 carbon dioxide and 12 NADPH2 are the end products. Then the place where they take place, glycolysis take place in the cytosol, citric acid cycle takes place in the mitochondria, and HMP shunt takes place in the cytosol. The number of ATPs produced in HMP are 0, in TCA cycle it is 10, and in glycolysis it is 9. Okay, these are single liner negative points of metabolism. So for example, TCA is not controlled by NADH. The metabolite which is not there in HMP shunt is glycerol 3-phosphate. Enzyme not used in gluconeogenesis is pyruvate dehydrogenase, that is the enzyme of glycolysis. Then brain and liver are not catabolized, cannot catabolize acetoacetate to carbon dioxide, water, and usable energy. Lactate is not formed in brain, it is basically part of skeletal muscle. Fatty acids are not utilized as energy source by brain, it uses ketone bodies and glucose. Then in humans, acetyl-CoA cannot directly give rise to formation of glucose. Acetyl-CoA is basically associated with the fatty acid synthesis or lipogenesis process. Acetyl-CoA is also a linking point between glycolysis and TCA cycle, that is earlier told. It is not a glucogenic substance. Then another important one, enzyme not acting in fasting to decrease glycogen, is angiotensin. Not a cause of fasting hypoglycemia is glucagon excess. Then increase in fructose 2,6 biphosphate does not occur when the liver glucose is short. Next table is showing the processes of anabolism and catabolism. So process of anabolism are glycolysis, synthesis of glycogen, triglyceride, fatty acid, protein, cholesterol, glycogenolysis, and gluconeogenesis. So synthesis of glycogen, glycolysis are anabolic in well-fed state, and glycogenolysis and gluconeogenesis are starting in the fasting and in case of diabetes mellitus. Coming to the catabolic pathways, fatty acid oxidation starts in the fasting and diabetes mellitus. Ketogenesis by non-hepatic tissue starts in the fasting and diabetes mellitus. Breakdown of fat starts in the either in fasting or in the diabetes. Coming to the high yielding points, uh, as earlier told, acetyl-CoA is the meeting or the linking point between the glycolysis and TCA cycle, and also in the metabolism of carbohydrate and fat metabolism. Then the first committed step in the glycolysis is by phosphofructokinase, and fumarate serves a link between urea cycle and TCA cycle. Cancer cell derives its nutrition from the glycolysis, and UTP glucose is the key substrate for glycogen synthesis. Coming to HMP shunt pathway. So hexose monophosphate shunt, also known as pentose phosphate pathway, there is a 0 ATP produced or consumed in HMP shunt. It is active in liver, RBC, testis, adipocytes, thyroid, adrenals, and lactating mammary glands. It is CO2 is directly produced in this pathway and never produced in the glycolysis. Okay, coming to some important points. In diabetes mellitus, all the genesis or synthesis and required enzymes are suppressed, except in gluconeogenesis and ketogenesis. The process here is ongoing. Lysis and catabolic pathways are promoted here. So the action of insulins are opposite. In case of hyperglycemic state, the insulin becomes active and it allows the breakdown of the product and get it consumed by the cells. The action of insulin are similar to metabolism in well-fed state, and metabolism is almost similar in fasting state and in starvation and in case of diabetes mellitus. In all these cases, the signal for the insulin gets activated and it promotes the metabolism of the carbs, fats, and proteins. It increases that in case of hyperglycemic or in case of a well-fed state. Okay. Next, the enzymes which are induced in diabetes mellitus in fasting state where there is low insulin and glucagon ratio. The three enzymes activated are glucose 6-phosphate, phosphophenol, pyruvate carboxykinase, pyruvate carboxylase. Okay, regarding insulin, as earlier told, its response gets activated in case of hyperglycemia and well-fed state. It stimulates to utilize and divert glucose, and it causes the genesis except gluconeogenesis and ketogenesis. The first response to hypoglycemia, in case of hypoglycemia, the amount of insulin secretion will be decreased. The action of epinephrine over the insulin is to decrease it. The insulin promotes utilization of glucose by muscles, mammary glands, adipose tissues, and myocardium by glucose transporters by GLUT4. Okay. The main regulatory hormones of insulin are glucagon, epinephrine, cortisol, and growth hormone. Next, the difference between insulin and glucagon hormone. Insulin is produced in response to hyperglycemia. Glucagon is produced in response to hypoglycemia. The two are secreted from the pancreas. In well-fed state, insulin gets activated. In case of catabolic state, glucagon gets activated. Insulin is inhibited by epinephrine, and its effect is making all kind of genesis except gluconeogenesis and ketogenesis. Okay, coming to GLUT or glucose transporters. So GLUT are different types. GLUT1 is found in the brain, renal tubules, placenta, colon, and RBCs, and it is independent of insulin. GLUT2 is found in the liver, pancreatic beta cells, small intestine, and kidneys, and also independent of insulin. GLUT3 is found in brain, kidney, and placenta, and is independent of insulin. GLUT4 is sensitive to insulin and is insulin dependent and is found in muscles, cardiac and skeletal muscles, and adipocytes. GLUT5 is a fructose transporter in small intestine and sperm. And regarding SGLT1 and SGLT2, SGLT is basically sodium glucose transporters. SGLT1 is found in small intestine and renal tubules, and SGLT2 are found in the renal tubules. GLUT2 deficiency or defect is associated with the glycogen storage disease, Fanconi-Bickel syndrome. Regarding glycogen metabolism, it is made and stored in the liver and muscle cells. The factors which affect the glycogen metabolism are epinephrine and glucagon, and it promotes glycogenolysis in the liver. Insulin inhibits the phosphorylase, inhibits the glycogenolysis in liver. Calcium affects only muscle glycogen metabolism, and it is stored in the muscles. Regarding glycogenolysis, the key enzyme is glycogen phosphorylase, and the common enzyme is glycogenolysis and glycogenosis. Here the cofactors are calmodulin, CAMP, and protein insulin kinase A. Glucose 1-phosphate is the end product in glycogenolysis. G6 phosphatase present in the endoplasmic reticulum of hepatocytes and not in hepatocytes in the liver and not in the muscles, and it promotes entry of glucose into the blood. Hormones which increases the adenyl cyclase and CAMP, that is cyclic adenosine monophosphate, are epinephrine, adrenaline, which is increased in the liver and muscles, and glucagon, which is in the liver only, and ACTH, which is in the adrenal cortex. CAMP increases the blood glucose by gluconeogenesis and glycogenolysis, and it increases the free fatty acid by lipolysis or increasing the hormone sensitive lipase. It increases basal metabolic rate, serum calcium, and increases the heart rate and contractility. CAMP inhibits glycolysis and cholesterol synthesis and inactivates glycogen synthetase, and thus it promotes the glycogenesis. So these are the carrier uh, molecule which are carrying the different active forms. Like ATP is carried in the active form of phosphoryl group. NADH, NADPH, FADH2 are carried in activated form electrons. Biotin as CO2, TH4 as 1C unit moiety, and TTP as aldehydes, lipomide as acyl groups. Hormones acting via CAMP are adrenaline, noradrenaline, glucagon, ACTH, TSH, PTH, gonadotropins, hypothalamic releasing hormones. Decreasing CAMP level uh, is associated with insulin and prostaglandins. Key enzymes and different metabolic pathways. This is an important cycle and commonly asked. So in glycolysis, the rate limiting step is phosphofructokinase. In glycogenesis, it is glycogen synthetase. In glycogenolysis, it is phosphorylase. In gluconeogenesis, it is pyruvate carboxylase. Curbs isocitrate dehydrogenase. In ketone body synthesis, HMG-CoA synthetase. Cholesterol synthesis, same, HMG-CoA reductase. Fatty acid synthesis, acetyl-CoA carboxylase. Catecholamine synthesis, tyrosine hydroxylase. Purine synthesis, PP ribose, P glutamoyl edimotransferase. Heme synthesis, ALA synthetase. urea cycle, carbamyl phosphate synthase, uric acid synthesis, xanthine oxidase, bile acid synthesis, 7-alpha hydroxylase. Coming to urea cycle, it takes place in the liver and the rate limiting step is carbamyl phosphate synthase. 3 ATPs are required in each turn of cycle. Kidney synthesizes ammonia from the glutamine. Brain synthesizes urea from citrulline. Excess of NH3 ammonia may lead to encephalopathy. Deficiency of urea cycle leads to hyperammonia. Hyperammonia inhibits the TCA cycle, tricyclic tricarboxylic acid cycle, depleting the ketoglutarate. OTC deficiency, OTC that is ornithine transcarbamylase deficiency causes hyperammonia type 2, the enzyme present here in the urea cycle. Hyperammonia type 1 is due to deficiency of enzyme carbamyl phosphate synthase that is present here in the cycle 1. HHH syndrome, that is hyperornithemia, hyperammonemia and hypercitrulinuria, is due to mutation in ORT1N1 gene that encodes for ornithine transporters. Gyrate atrophy of retina is due to mutation in ornithine D aminotransferase. Okay, coming to lipoproteins. So the different lipoproteins are chylomicrons, VLDL, LDL and HDL. Very low density lipoproteins, low density lipoproteins and high density lipoproteins. So mobility in the electric field is least for chylomicrons and highest for the high density lipoproteins. Apoprotein presenting E's are different. So remember this, these are asked like ApoB48 represents chylomicrons, ApoE represent chylomicron and VLDL and LDL also. Next, uh, Apo beta 100 represents LDL, Apo A1 and 2 represents HDL. Their synthesis, chylomicron takes place in intestine, VLDL in the liver and intestine, and LDL by degradation of VLDL it is synthesized in the blood and liver of VLDL. HDL is present in the liver. Okay. The size of chylomicron is maximum. The size of HDL is minimum. Then density is lowest for chylomicron and maximum for HDL, high density lipoproteins. Then major lipids formed from these are triglycerides. VLDL also form triglycerides and LDL forms cholesterol. The function of chylomicron is transport exogenous and dietary triglycerides. Again, it is also responsible for transport of triglycerides. LDL are atherogenic or responsible for coronary artery diseases. HDL high density lipoproteins are scavenging action, transport cholesterol from peripheral tissue to the liver for further degradation. Coming to hyperlipoproteinemias, so the genetic disorder of exogenous hyperlipemia is because of elevated chylomicrons, elevated triglycerides. The patient here presents with the eruptive xanthomas, lipemia retinalis, hepatomegaly, pancreatitis, but no atherosclerosis. Here you will get ApoC2 or lipoprotein lipase deficiency. Next is hypercholesterolemia or Mueller-Harbit's disease. It is the most common type. Elevated are LDL, low density lipoproteins, and elevated lipid is cholesterol. Patient presents with xanthelasma, arcus sinalis, tendon xanthomas, and atherosclerosis. The mutation is in the LDL receptor mutation. Next is combined hyperlipidemia, which has elevated LDL and VLDL, so both triglyceride and cholesterol are elevated. Patient present with the palmar and tuberoeruptive xanthomas and atherosclerosis. Pathophysiology is VLDL overproduction. Next is remnant hyperlipidemia, which has elevated beta VLDL and chylomicrons. Elevated lipid are again triglyceride and cholesterol. It present as atherosclerosis. Enzyme deficiencies of apoenzyme E. Next is endogenous hyperlipidemia, which has elevated VLDL. Triglycerides are raised. Glucose intolerance is present, hyperinsulinemia is present, type 2 diabetes mellitus, obesity and atherosclerosis are present. There is overproduction of VLDL lipoproteins. Next is mixed hyperlipidemia, which has raised VLDL and chylomicrons. Both triglyceride and cholesterol are raised. Patient present with pancreatitis, xanthomas, but no atherosclerosis. Again, that is also because of over